Welcome everyone to the webinar. We will get started in just a couple minutes. Welcome everyone. I'm excited to see the attendance here. We'll get started in just another minute. All right. Hello, and welcome to the second installment of AFTO's Fall Webinar Series. I'm your host, Carrie Rigdon. Today we're featuring an important presentation on Salmonella dry biofilms and adjusting your sanitation practices to target these biofilms effectively. We have an excellent speaker here with us today, but first a little housekeeping. So the session is being recorded, and all of those who registered will receive a link to the recording as well as a copy of the slide deck in the next few days. If you have any questions for our speaker, please leave them in the Q&A box, and we'll address those during the question and answer portion at the end of our session. We ask you to use the Q&A tool instead of the chat. That just makes it easier to see the questions. Also, if you'd like to ask questions verbally during the Q&A, you can use the raise hand feature at the bottom of the Zoom screen, and we'll prompt you to unmute. Now, it's my pleasure to introduce our speaker. Jean-Yves Maillard is a professor of pharmaceutical microbiology at the School of Pharmacy and Pharmaceutical Sciences at Cardiff University in Wales, with over 28 years experience in the field of biocidal product efficacy and microbial resistance. He has a particular interest in novel disinfection technologies, sporicides, complex hydrated biofilms, and dry surface biofilms. Over the years, he's been the recipient of multiple awards, including innovation and engagement ones. He's published over 200 peer-reviewed articles, 20 book chapters, 3 books, and over 150 refereed conference communications. Professor Maillard is a member of the editorial board for the Journal of Antimicrobial Chemotherapy and is a chair of the British Standard Institute Group on Antimicrobial Hard Surface, a member of the TC 330 Committee on Antimicrobial Hard Surface, and lead TC 330 Working Group 1 on assessing the risk of microbial surfaces to antimicrobial resistance. Now I'd like to turn it over to Professor Maillard.
Well, thank you very much, Carrie, for that introduction, and welcome to the audience to this webinar. I'll start first with it works, here you go, a disclaimer. So some of the results I'm going to present during these presentations are from a project supported by Procter & Gamble and funded through Cardiff University, so in Wales. I'm also the director of Biocide Consult. But on this note, I have nothing pertinent to a mention about this presentation. So as the start, I'm going to talk about dry surface biofilms. All right. Dry surface biofilms, they're widespread on environmental dry surfaces. They may contain pathogens. They're not visible to the naked eye. They cannot be detected by wet environmental swabbing or contact plates or any other means when the surface is dry. And they can survive to high concentrations for a very long period of time in a dry environment. They are more resilient to environmental disinfections. And effective eliminations of those dry surface biofilms usually mean a combination of an effective formulations, but also of mechanical removal. Salmonella, enterica, and Listeria do form dry surface biofilms. So all those key points and the key facts, I'm going to try to demonstrate those key facts for you during this webinar presentation. So at the end, hopefully, of this webinar, you will be able to make your own mind about those new type of biofilms. So when we start talking about biofilms, we need to start from the start about what now we call, or what I call, the hydrated biofilms. So biofilms are very ubiquitous in nature. You can find them absolutely everywhere. But when people think about biofilms, they're thinking about those hydrated biofilms, those mass of microorganisms you can find, for example, in the oral cavity, but also in drains, for example, or in different factory settings. They are visible, right? And those hydrated biofilms have been known for quite a number of years now, and we know a bit more about how do they form on surfaces. Usually they help with the conditioning film on surfaces that help the attachment to your microorganisms. That stage is reversible. And when they're starting to colonize the surfaces, they suddenly change their properties. And that enables them to suddenly start proliferating, but also to make a matrix of a exopolysaccharide, so a mix of polysaccharide, extracellular DNA, but also proteins. And that biofilm is going to grow and become mature. And when you've got a biofilm mass, then the microorganisms in that biofilms are going to start spreading around that biofilms, either as individual free-floating bacteria or as a biofilm raft that can go and colonize other surfaces. Now, why biofilms usually are very important to consider is because by itself, a biofilm is a structure that will cause us problem in terms of antimicrobial effect, whether it is with a chemotherapeutic antibiotics or whether it is with a disinfectant. And recently, we've published an up-to-date review on the mechanisms really involved in biofilm resistance to any antimicrobials. And there are many mechanisms involved in those hydrated biofilms to enable them to survive a disinfectant. So, for example, we have a mechanical quenching or neutralization in that biofilm, especially because of that matrix. We have in that biofilm some microorganisms that are less metabolically active, and some of them are rather dormant, and some of them are called persister cells, and they're probably responsible for biofilm regrowth following treatment. Within that biofilms, you've got also lots of communication through chromosomes, lots of gene exchange, on the expressions of mechanisms and enzymes that are going to enable the biofilms to deal with any stress molecules, and particularly antimicrobials. In addition to that, in those biofilms, you've got an increased mutation rates that enable those microorganisms within the biofilm to survive better. So overall, biofilm is really problematic, especially when we talk about controlling biofilms we do not want. And in fact, when we're looking at the susceptibility of hydrated biofilms to an antimicrobials, we know for a fact that those hydrated biofilms are far less susceptible than your free-floating bacterium, for example, your planktonic cells that floating in suspensions, but also they are far less susceptible to those cells that are attached to a surface. And there are many publications that have shown that, for example, in hydrated biofilms, usually your biofilms is at least 1,000 times less susceptible to antimicrobials than your free-floating microorganisms. So that really matters. Now, of course, in your biofilms, lots of them are complex biofilms. So that means they contain many microorganisms. They can contain a mix of bacteria, fungi, viruses, protozoa. And some of them contain only bacteria. And usually the one that we find infecting humans and associated with an organ, they are monobiofilms. But also the resistance of biofilms to an antimicrobial will depend on the type of microorganism you have in that biofilms. But by far, those hydrated biofilms, they are complex biofilm, containing multi-species. And I'm lucky enough to head a big European research group on complex biofilms in the One Health area. So that means in hospital, domestic, on the animal industries. And really, when we look at the biofilm, we find, particularly in drains, it's a very complex screen. You've got lots of blobs, lots of different colors. On the left-hand side, you can see biofilm from a pig farm, in the middle, a home, and at the bottom from hospitals. And all those little blobs correspond to different biofilms. All those lines you have on the left-hand side corresponds to different biofilms. So basically, just that pictures tell you how complex the microorganism, in this case, just the bacteria we have in those biofilms are. And we can also see that depending where your biofilm is from, the diversity of that biofilms, what the bacteria they contain in that biofilms will be different. Right? So those are about hydrated biofilms. But today, really, I want to make the parallel with those new type of biofilm called environmental dry surface biofilms. This is quite a new science, and the first time really the term dry surface biofilms was used with my colleague in Australia, Karen Vickery, in 2012 in that papers there on the left-hand side, the first time she mentioned the term dry surface biofilms. And since then, that's really a new concept of having a dry biofilms on a dry surface, which was quite novel at the time, has become really interesting and has certainly grown quite a lot in number of publications and interest worldwide. Now, most of the dry surface biofilms data we have so far concern the health industries. But there are lots of parallels between healthcare and also the food factories or the food preparation area when we thinking about the presence of those biofilms on dry surfaces. And in fact, recently, last year, we published the first paper on actually food pathogens forming dry surface biofilms on the response to disinfections. Now, the interesting part is what you might want to ask is what really are environmental dry surface biofilms? Well, the samples we have, mostly, as I said, from healthcare industries, are very complex microbial communities on surfaces. They usually do not cover the entire surfaces, but there are raft of biofilms scattered around your surfaces. But those are really viable microorganisms. They contain a range of microorganisms from your environmental microorganisms. So if we talk about bacteria, mostly you're going to have, for example, bacillus species, but you can have also your skin microorganisms, especially in healthcare. You're going to have all your staphylococci, for example, these biofilms. And sometimes in those biofilms, you find your pathogen. You find your MRSA, if you're in healthcare. We find the E. coli, the CPE as well, embedded in those biofilms. But usually, you find mostly gram-positive bacteria, particularly bacteria that can survive desiccations very well. So we know about those dry surface biofilms. We know they can colonize various types of materials, so hard surfaces from porous surfaces. We find them actually everywhere. When we look for them, we find them absolutely everywhere. Now, one of the major differences between those dry surface biofilms and the hydrated biofilms are those dry surface biofilms' depth is about only 10 to 30 microns. So you cannot see them with your naked eye. So you don't know they're there, and that's an issue. And that's the difference between a dry surface biofilm on dry surfaces on your wet mass biofilm you find, for example, in the drains. But very importantly, and that's the distinction I'm going to make from the start and throughout that presentations, the dry surface biofilms, there are not hydrated biofilms that have been dried on the surfaces. There are biofilms that exist on that form on their own accord. So when we look a bit more at sampling the environment, and again, those data mostly concerns the healthcare industries, Personally, when we've done that work, we find those dry surface biofilm on 90 to 95% of the surfaces we sampled. Right? On those surfaces from the healthcare industries, usually they're coming to my lab, they've been cleaned, usually also disinfected with a sodium hypochlorite. On what we do in the lab, we check that the surface contains any microorganism, and usually they do not. And we do that by swabbing, the same way that environmental monitoring is being performed in practice, but we can't find anything. And then we've got a way to look for those biofilm, and we find them, as I said, to 90, 95% of surfaces. So we know they are there in the environment, and we certainly believe they do contribute to pathogen survival, particularly in healthcare settings, despite cleaning and disinfections. As I said, one of the main issues we have at the moment, they cannot be detected by your normal environmental monitoring. So how do they form? Well, still at the moment, it's a bit of a theory how they form. Do we need a conditioning film on the surfaces? We don't know. We believe these dry surface biofilms are biofilms that detach themselves either from a person or they're associated with food, for example, or any spillages you would have on that surface. Is it reversible when they touch the surface? Do they stay there and start forming a dry surface biofilms? We don't know. Limited colonization, that's for sure. We know that it's limited, and we know they're forming raft of biofilms scattered around the surfaces, but not covering the entire surfaces. What is very clear to us is the environmental conditions are very harsh. It's desiccation, so it's not very prone to the growth of the microorganisms. But we believe the biofilm expanding every time you've got a wet phase. For example, when you clean the surfaces, when you spill something on your surfaces, then you might have the growth of those microorganisms. We don't know much about dispersion of those biofilms, but we know that when the biofilms, those dry surface biofilms are being disturbed, they're easily transferable, for example, by touching. Now, very interestingly, still with the healthcare area, that the dry surface biofilm we find in environmental surfaces, we find exactly the same way in medical devices, and particularly endoscopes. And those colleagues in Canada published a very nice paper looking at different formation of the biofilm between hydrated biofilms and what they call the cyclid built-up biofilms, which are dry and desiccation. And in the model is an accumulation of microorganisms on the surface, but those microorganisms, they just not accumulate, but they also grow. And very interesting for us is when we look at scanning electron microscopy of the lumen of endoscopes, when you've got microorganisms, we find exactly the same structure in those lumen that we find when we look by electron microscopy on the surface of environmental surface, for example, in the hospitals. Now, why biofilms, dry surface biofilms are important is we will show later on that they are also very resilient to disinfections. Now, in terms of mechanisms of resistance of biofilms, they will be quite different with the hydrated biofilms. There are some small similarity, like the productions of that extrapolar substances, so that matrix of biofilms. The difference in hydrated biofilm, they're producing loads of EPS. In dry surface biofilm, because of the nature, far less. At the moment, we don't know if we've got a extracellular DNA in those dry surface biofilms. We won't have a depth gradient because dry surface biofilms are only 15 to 30 microns on the surfaces. But very importantly, desiccations play certainly a role in their lack of susceptibility to antimicrobials. They certainly have reduced metabolisms, and we're not sure yet they've got persister cells. So when now we've got those dry surface biofilms, telling people that, yes, there's an issue, it's not good enough. We need to find really solutions to be able to eliminate or control those dry surface biofilm from surfaces, particularly when those dry surface biofilms contain pathogens. So when we're looking at protocols available to look at the effect of antimicrobials against microorganisms, whether biofilms or not, then we've got different protocols available. when we're talking about planktonic suspension, so bacteria in suspension, or bacteria just dry on the surfaces. Then if we're looking at antibiotics, then we know we've got, well, at least with suspension, MIC determinations, like broth dilutions. If we're looking at biocides or disinfectant, we've got suspension test. When we're looking at bacteria dry on surfaces, we've got carrier test, and we've got plenty of EN test in Europe. You've got plenty of ASTM and ISO test otherwise. And interestingly, at least in Europe, we've got now a product test, which really not testing only the formulations, but also the delivery systems. For hydrated biofilms, it becomes more complicated. They've got far fewer methodologies. So in the US, you've got few standards to look at hydrated biofilms, and you like usually Staph aureus or Pseudomonas aeruginosa hydrated biofilms. There's no product test really for those biofilms. But when we talk about dry surface biofilms, then there's no standard. We have some companies have used academic methods, including the ones I'm going to describe in a minute. But what is absolutely clear, and I want to make that point very strongly, is there's no point of testing a Pseudomonas aeruginosa dry surface biofilm because it cannot exist. Pseudomonas aeruginosa doesn't like desiccation, and it will die very, very quickly. So in terms of susceptibility of your dry surface biofilms, it adds a level of insusceptibility, of resistance. So you've seen that slide 5 minutes ago, talking about the different susceptibility between the planktonic cells attached to the surface, your hydrated biofilms. Your dry surface biofilm is even more resistant than your hydrated biofilm. So I will show you that through that presentation in a second. Very importantly as well is how long can a dry surface biofilm survive on surfaces. Unfortunately, I do not have data on Salmonella or Listeria just yet. I've got data on other microorganisms, data that's coming from my lab. At the moment, we're working a lot on vancomycin-resistant enterococci, and you can see on the right-hand side that those VRE can last to a very high number over 44 weeks. And that's incubation at 20 degrees and at a relative humidity between 40 and 60% humidity for that length of time. And we still recover a very high number of microorganisms. Again, that's confirmed that for some species that form a dry surface biofilm, they can persist on the surface for a very, very long period of time. On the left-hand side, you've got a graph with other species. On the top, those are a Bacillus, a Lichenoformis, or a Bacillus subtilis, an isolate from an endoscope wash disinfector. And those two isolates produce quite a large amount of exopolysaccharide that you will also see on the dry surface biofilm. And you can see that I survived to extremely high number within 6 months. Other Bacillus, which is a standard strain, survive less well on surfaces. On your Staphylococcus aureus survive up to 4 months as a dry surface biofilms on the surface. So let's talk now about dry surface biofilms in the food industry. We know we've got an abundance of dry surface biofilms on surfaces in food processing plant. Well, sorry, the abundance of dry surface biofilm in the processing plant at the moment is unknown because we haven't looked specifically for dry surface biofilm in those industries. The exact composition, again, on DSB is not known because, as I said, usually when the surface is dry, you cannot really pick up any dry surface biofilm by swabbing. And I know the food industry like the contact plates. The prevalence of resistance to disinfection of dry surface biofilm in food industry has not been really investigated. At the moment, we're the only group worldwide who has done that so far, and I'm going to present some of the results today. So how do we test for dry surface biofilms? Well, we had to find really a method to form those dry surface biofilms in a way that is reproducible, so we can test a different disinfectant on different products against those dry surface biofilms. And the way we do that is to alternate a microbial growth between a wet phase and a dry phase over a 12 days period. And by alternating a wet phase and a dry phase, and by adding some organic load to that dry surface biofilm regularly, we end up at the end of 12 days a very consistent dry surface biofilm that resemble very well the dry surface biofilm we find on environmental surfaces. And with using that protocol, we've been able to form dry surface biofilms with a very wide range of microorganisms, and you can see including Salmonella and Listeria as well. Now, we've been working on dry surface biofilms for a number of years, as I said, mostly at the start for the healthcare industries. And what's become very clear to us how to control those dry surface biofilms is in two ways. And for us, a product needs to do two things. The first one is to reduce the number of microorganisms on the surfaces. The second one, to stop the transferability of those dry surface biofilm from that surface, whatever is left after the disinfection process, to other surfaces. As I said to you earlier on, what is clear with the dry surface biofilm is when the surface is dry, it's not transferable. You cannot pick it up with wet swab or contact plates. When the surface is disturbed, usually following, for example, a detergent, some colleagues done that in Australia, then the microorganisms become transferable. So you imagine that when we're going to use a disinfectant or a cleaning agent or combinations of cleaning and disinfection, we might disturb those biofilms. So very importantly, in my view, is we need to look at two factors, the log reductions and the transferability. And in fact, we've done that on many occasions. So those ones with results with Staph aureus, a dry surface biofilm, and you can see here on the right-hand side column, the log reduction on the surfaces on the transferability. In green boxes, that means it passes the test. So that means we achieve more than 4 log reductions, or 99.99% reduction on the surface. And in terms of transferability, we've got less than 100 bacteria transferred post-treatment. On the left-hand side, then you've got the different agent and the concentrations. But what should be very clear with that table is the dark blue agent is with the medium of a wipe, the wipe becoming very important. And when you've got a combination of your disinfectant on the wipe, then you're starting to have a product that works against the dry surface biofilms. Not always, but in general. When you've got an agent without wiping, then it doesn't work. Now, the wiping test we've used in combination with our dry surface biofilm is inspired by the ESTM 2967 tests called the Wiperator test. And if you're not familiar with that test, it's an STM method. It's using a machine called the Wiperator, which control the weight imparted on the surfaces, but also the wiping movement as well. The contact time is multiple of 5 seconds, but it is a very reproducible way to wipe a surface. So it gives us some indications on some relief that when we're using that devices, we know how to control the disinfectant on the wipes. And we've published that with my colleague Said Sattar quite a while ago, that all the factors that can impact the efficacy of wiping. So if we apply that to controlling dry surface biofilms, from what we know and from all the data we accrue here in Cardiff, but also the data from my colleague Karen Vickery in Australia, then it is absolutely clear that, as I said, measuring transferability and recovery DSB post-treatment is very important, that we know that most disinfectant against a bacteria that usually matter in healthcare, most disinfectant cannot prevent dry surface biofilm transferability. And all the results so far we've produced really correlate with all the publications, well, the few publications that have been done so far. So how does it look with Salmonella? And as I said, we're fortunate enough to be the first group to really look at Salmonella enterica dry surface biofilm. So Salmonella from very nice dry surface biofilm after 12 days, very consistent biofilms. And here, that's a paper we've published this year. And you can see in the table where here we compare for you some indication between the dry surface biofilms and the hydrated biofilms. Again, remember I told you the dry surface biofilms is usually far more resilient than your hydrated biofilms. So if we concentrate, I don't know if you can see my pointer, if we're looking at dry surface biofilm, the second line without wiping, to compare directly with the hydrated biofilm without wiping, you can see that in terms of log reductions we have with those products, then they do not work. But you can see that with the hydrated biofilms, you can still remove far more microorganisms than you do with the dry surface biofilms. Now, to control appropriately your dry surface biofilms, your Salmonella dry surface biofilm with the product, then you need to have wiping. You need to have, in this case, a 5 seconds wiping with a weight of 300 grams. And I think we had a 1 minute residual time at the end. So what about percentage transfer? Again, I said to you, it's very important to look at transfer as well. Again, you can see that, again, the product used appropriately with wiping were able to stop the transfer of those microorganisms from that surface that's just been treated. Now, we can go further than that on looking at far more formulations. Again, that's in that paper we've published this year. Again, 2 columns, the log reduction from the surfaces, and then the percentage transfer on the right-hand side. In red, again, that means it doesn't really work very well. At least we've got less than 4 log reduction on the surfaces, and we've got then more than, in this case, 50, more than 50 positive contact in terms of transfer. So you can see again here that usually, in a nutshell, without wiping, your product doesn't work as well. So if you just add the product and leave it there, it doesn't really affect the dry surface biofilms. When we've got wiping, then you're starting to have a product, not only that can reduce your Salmonella with more than 4 log reductions, so by 99.99% on the surfaces, but also prevent the transfer of whatever Salmonella left on that biofilms to other surfaces. So more recently, again, those are data I'm going to ask you not to share. Those are new data that are going hopefully to be submitted for publications in the next few weeks. Again, we've done even more work. And in this case, what we look at in this case is not only the formulations, but the type of material you use for wiping the surface. So in this case, for all those different histograms you can see on those graphs, those correspond to a formulations in associations with different materials. And usually it's a spray. So you spray the surface with that formulations, and then you wipe the surface. The wiping in this case was done with the ASTM standards. Now, on this graph, again, we've got two sets of data. The first one, the blue bar, corresponds to your Salmonella enterica planktonic suspension dry on the surfaces. The gray bar corresponds to your dry surface biofilm of Salmonella. Right? Early on, I said to you that dry surface biofilms were far more resilient, so far less susceptible to just planktonic cells dry on surfaces. Here you evidence. Here you can see that when we've got the planktonic Salmonella suspension that has been dry on surfaces, you can see that that formulation combined with any of those different materials work. On the right-hand side, you've got product A on product B, those are already made product. And you can see, again, those bacteria dry on the surface, they work. When we're looking at the gray bar, those are your dry surface biofilms. You can see the reductions on the surfaces is certainly not 4 log reduction, but for one product. It averaged between 2 logs and sometimes less than 1 log product. Now, interestingly, in that graph, where you've got a very low bar, where you've got a very low gray bar, so for the material 2, for example, on the formulation on for your product, the material 2 has been soaked in the formulation. So it's not a spray on the surface, it's been soaked in the formulation. So it behaves the same as a product A and B. So it's not very efficient in controlling your dry surface biofilms at the end of the day. Another set of data we have in this case is again a difference between different Salmonella, because we were interested is if we've got a standard strain of Salmonella, which is your SL1344, and if we've got more environmental food industry strain of Salmonella, do they behave the same to that product? And you can see that roughly they do behave the same in this case. All the Salmonella we tested do behave the same. There's no other problems. Now, the material 6, which worked better, so in the previous slide on this one, that is a sponge type of material rather than just a different type of other materials. And that's certainly make a huge difference in removing your dry surface biofilms. Also, what you can see on this slide in blue is just using water with your formula, with your materials, in gray as using the formulations, which is the quark-based formulations on the materials. And in most instances, against those dry surface biofilm, you can see that whether you use the water or that formulation you've got the same results in terms of reductions of bacteria on that surface. Now, the difference we've seen is Listeria monocytogenes. With Listeria, all the data we have that hopefully, as I said, going to be sent for publication in a couple of weeks' time or in a few weeks' time, is Listeria is far more resilient as a dry surface biofilms than Salmonella enterica biofilms. Far more difficult to control on surfaces with the same product, with the same materials. Now, where things are starting to be interesting and where we're starting to see really differences between using a formulation or just water with the materials is when we're looking at transfer. Again, as I said, to control properly a dry surface biofilms, we need to look at not only reduction on surfaces, but also at the transfer post-treatment. And when we're going to see differences, and we're starting to see differences, not always with all the microorganisms, is when we've got a formulation. Usually when you've got the formulation rather than water, we're going to transfer less microorganisms. Here, it is quite not that clear when we're using just one wiping post-treatment. So we're spraying and we're using one wipe. On the right-hand side, that graph is we spray and then we're using 3 different wipes to wipe the surfaces. And here you're starting to see quite a lot of differences in terms of effectiveness of your formulation comparing to water. The same with another Salmonella enterica. In this one, big difference between our standard strains on that environmental isolate. That environmental isolate, when you use your formulation A, you do not transfer microorganisms. So if you remember previous slide, it was not necessarily very good at removing the bacteria from surfaces. We had only roughly about 2 logs, 99% reduction on new surfaces, but at least the formulation will prevent that transfer. So really briefly in sort of conclusions, dry surface biofilm in the food industry, as I said at the moment is, yes, I believe you're going to find dry surface biofilm in the food industry. I mean, when you're looking at production plant, if you're looking even at the, I guess, food processing as well, is obviously going to have residues of food. Even if you clean and disinfect, you're going to have those food prone to dry surface biofilms. So comparing dry surface biofilm in the food industry with what we know about healthcare, we know in healthcare they're very widespread because we've done the work. On our results in the UK are very similar to the results they've got in Australia and elsewhere in the world, that we find those dry surface biofilm in 90 to 95% of surfaces we sample in healthcare. We know they are persistent. We know they're not easily detectable, certainly not with your current swabbing or contact plates, but we can recover them nevertheless with other methods. When they are disturbed, then become transferable. We know they are more difficult to control than, for example, hybrid biofilms. And as I said, we know they're transferable. So what about for the moment with the food industry? With the data we've got in the lab, we know they are more difficult to control than hydrated biofilms, and we know they are transferable. At the moment, what we're lacking is the evidence that we find them in the food industry, but I believe we would find them, that's for sure. So what do we have overall? So what hopefully through that presentation I was able to articulate is, well, in terms of food isolate, yes, Salmonella on the Listeria food can form dry surface biofilm on surfaces. They're able to resist desiccations. We can recover them easily on their pure, very variable microorganisms. As with the dry surface biofilms in the healthcare industry, they are not visible to the naked eye. They are more resilient to environmental disinfection. So the work I presented here today about Salmonella on a Listeria dry surface biofilm, hopefully I managed to convince you they are quite resilient in terms of exposure to a disinfectant. That effective eliminations required not only an effective formulations, but also mechanical removal. And I cannot stress out that mechanical removal so far has been absolutely key to control those dry surface biofilm on surfaces. Hopefully, I managed to articulate that really for me, when we look at controlling those dry surface biofilm on surfaces, is we need to look at two parameters. We need to look at reductions of those biofilm from surfaces, but we need to look at the lack of transfer of those microorganisms after treatment. As I mentioned, is usually those dry surface biofilm, when dry, they don't appear to be transferable, but when they're being disturbed, they are. And finally, hopefully what I managed to articulate as well is with the latest results, which, as I said, hopefully will be sent for publication in a few weeks' time, is the type of material used for wiping in association with your formulation matters. And that's something we've known for a long time when we started working on dry surface biofilms, is at the start, again in healthcare, those combinations of material and formulation were very important. And we found in the healthcare setting for those dry surface biofilms that if you had the wrong formulation with the wrong material, you might make matters worse. So I think for an academic point of view or for an innovation point of view, there's still some lots of scopes to really look for better combinations between the right formulation on your right materials that will enable you really to control those dry surface biofilms. True, at the moment, what really is missing is evidence. They are in the food industry, but I have no issues, or I'm pretty confident to know that we will find them when we look for them. But the important things for me is, I know they're over there, but what do we do to control them effectively? So again, appropriate formulations on the appropriate combinations with the right material to eliminate those dry surface biofilms. So that was quite a whistle tour about the world of biofilm and particularly dry surface biofilms for the food or relevant for the food industry. And of course, I need to thank some absolutely fantastic people that worked for me over the years, are still working for me, that have been absolutely essential to do all those work and all this data that we managed to publish over the years. Me, I'm easy, I'm in my office and I'm just telling them what to do, but those colleagues there, they're all doing the groundwork, the lab work to present to you all those results. So thank you very much for listening to me. So we've got now quite a few minutes really to answer any questions you may have. Thank you.
Excellent. Thanks. Thank you, Professor Maillard. And clearly it is, this is kind of cutting edge in terms of there's still a lot unknown. So if you have, we have a few questions in the Q&A tool. If you please feel free to put your questions there, or you can raise your hand and Amy will help get you unmuted. So The first question we have is related to that slide that you showed about the time a biofilm can survive. So the question is really about um, just some assumptions around that. Is that assumption about how long it can survive? Is this as long as the pathogens have optimal conditions for survival, like pH, temperature, substrate, etc., or is this with kind of minimum quality of these conditions?
Yeah, I mean, it's not in ideal conditions. I mean, we put them just on the surfaces. What they had is they had a bit of proteins or organic load to survive when we make the dry surface biofilms. And then we leave them on the surface. They've got nothing else, nothing at all. We put them in a box with a control relative humidity at 20 degrees on nothing else. So they're very poor conditions. Suddenly after 44 weeks using or 6 months, so very, very poor conditions. So they will survive very well. And I believe is, from what I've shown you, is some microorganisms do form loads of extracellular polymeric substances that suddenly protect them from desiccation, so they enable them to survive to a much longer time. But it also depends on your microorganism. I mentioned the vancomycin-resistant enterococci. Those ones do form an exopolysaccharide, but not to huge extent. So they're just purely the nature of the microorganism that enable to resist desiccations. So in terms of gram-negative, in hospital Klebsiella pneumoniae, very happy to survive a long time, happy to survive desiccation. Salmonella and Listeria, we haven't done yet a long-term study, so that we're planning certainly to do that to see how well we can find them on surfaces. But again, those are very minimal conditions. There is no additional food, additional water. They're just here at 40–60% humidity with nothing else for a period of time. So it really depends on the intrinsic ability to survive those very harsh environments.
Great. Yes, thank you. And we have a second question about salmonella and antibiotic resistance. What do we know about that right now?
Well, we know that some they can have some antibiotic resistance genes as well. So some of them can be resistant to antibiotics and difficult to control, that's for sure. Now, if they form dry surface biofilms, having an antibiotic resistance genes or expressing an antibiotic resistance mechanisms will not stop them forming dry surface biofilms. So if they form dry surface biofilms, whether they are antibiotic resistant or not, they will form a dry surface biofilms. And in parallel, we know that with a pathogen from healthcare industries. As I said vancomycin resistant enterococci, so they're resistant to vancomycin on other antibiotics, but we've got also strains that are not resistant to vancomycin. They form also a very nice dry surface biofilms. That's not a real issue.
I don't see other questions in the Q&A, but I actually had a question. What I was really intrigued and maybe a little concerned about is that here in the US to actually do an environmental assessment of is there salmonella in this processing facility or this restaurant, et cetera. The method that we use is we have sponge swabs, so swabs on a stick. We put in some de neutralizing broth and then do our little pattern. From what you know right now, or maybe it's not known, but from what you know right now, is that sufficient to actually recover a salmonella that might be forming one of these biofilms, or is that one of the big questions?
No, the answer is no.
Oh, the answer is no.
Yeah,
Oh,
no,
so even if,
because exactly what we do in healthcare industries,
okay.
exactly the same. So wet swab on a dry surface, put that in neutralizing broth, and you've got recover absolutely nothing. So all the environmental surfaces we have from healthcare, that's exactly what we do. We do a control first to see if we recover any microorganisms. And when we're confident we don't recover anything, then we look for dry surface biofilms, and then we find them. But as I said, when they're disturbed, on that work we've done, but also my colleague Karen Vicker in Australia, is when they're disturbed, for example, with the detergent, then they're becoming transferable. So for a healthcare industry, what would be interesting to do is immediately after you've done your detergent, try to recover your microorganisms, and then you might pick them up. But again, in food industry, when you do your environmental sampling, I don't know if you do it immediately after detergent when the surface is still wet, or if you do it randomly during the day, regardless as to when your cleaning or disinfection process is. And that will make a difference, I believe. So at the moment, which I haven't mentioned, we do work on where to try to identify those dry surface biofilm from surfaces. At the moment, what we have is a destructive way, which is not very good. But we're working on the way to try to look for those dry surface biofilm on surfaces.
Wow. Yeah. So that's definitely something we're going to have to watch out for, because that's,
Yes.
that's the mechanism that we use to do that sampling. So I know we've got just a minute or so left. I've got a couple questions. One is question about iodine as one of the formulations used as a disinfectant. Uh, any insight on that? They didn't see
We haven't
iodine as
we haven't used
yes.
iodine. I don't think I recollect any publication using iodine on any dry surface biofilms. But what I can tell you is most probably I probably would not really well guess, but what I believe is, again, with its iodine, but without mechanical removal, which, as I said, is absolutely essential to eliminate dry surface biofilm. So iodine on its own, without mechanical removal, I believe that it's not going to do much.
Okay. And then Amy informed me we have a raised hand. Amy, can you help us out with that?
No, never mind. Oh, sorry, it is back raised. All right, Maureen, I am going to allow you to talk. You may have to unmute yourself, so go ahead and unmute yourself.
Hi, this is Dr. Molo. I love bugs, and this webinar just highlighted the things that I enjoy most, their ability to adapt and do some very fancy things that we typically don't expect. So I am a food safety professional, and just listening to this conversation made me think about dry manufacturing and the fact that we typically don't want to make surfaces wet. So lots to think about. I don't have a particular question. Dr. Mayad, I just wanted to acknowledge that this was very, very fascinating for me as a scientist. So thank you for making the time.
Oh, thank you for your comments.
Excellent. And that kind of does bring us to close. If you still have questions, you can reach out to afto@afto.org, and we will send them to Professor Maillard. Just to closing comment, thank you for your participation. A lot of great insightful questions. Please keep an eye out for the post-webinar email that will include access to this recorded session, as well as a PDF version of the presentation and additional resources that our speaker would like to share. And remember to check out the rest of our Fresh Take Fridays, the fall webinar series. This has been very successful, and we still have more, and you can register for them. Let me just put that into the chat real quick. Oops, I want to send that to everyone. Okay, send that to everybody. Great. And with that, thank you very much, and really appreciate it. Enjoy your rest of your day.
Thank you.